All right, so deep dive today. We're going to be tackling population genetics. And now, as a philosopher, you might be thinking, well, this is kind of a hardcore science topic, right?
Yeah. Mm-hmm.
But we've got some sources here, mainly this big entry in the Stanford Encyclopedia of Philosophy, all about population genetics. And it turns out it's chock full of philosophical stuff.
Oh, yeah, for sure. I mean, you're talking about inheritance and variation
Yeah.
and how evolution really works, right? Like down to the nitty-gritty.
Exactly. And the Stanford Encyclopedia, they make this point, to really grasp evolution, you got to get this population genetics thing, especially if you're going to be doing philosophy of mind, ethics, any of that stuff.
Totally. It's like the fundamental toolkit for that. You can't just stay with the broad strokes of evolution. You got to go deeper.
So we're going beyond the basics here, down to the genetic level. But it's not just about genes, right? There's more to the philosophical angle.
Absolutely. Population genetics, it's like a perfect case study for philosophy of science, right?
Yeah.
It gets you thinking about how we build scientific models, the whole idealization thing, how we explain stuff in biology, and of course, how theory and all that data, all the data we have now, how those work together.
So basically, understanding population genetics, you're not just learning about genes and how things evolve, you're learning about how science itself works, which is pretty cool.
Exactly. Plus, you can't forget all the debates within philosophy of biology itself. Stuff like, how does evolution cause things? How do we explain how creatures adapt through selection? And what about chance? Does random stuff play a big role in how life became so diverse? And even, like, what's the right level to be looking at when we study biological systems? So this deep dive should give you the basics so you can jump into all these juicy philosophical discussions about evolution.
Sounds good to me. So where do we start? I guess, historically, how did this whole population genetics thing even get going?
Well, like our source reminds us, Darwin, back in 1859, he published On the Origin of Species. And in it, he had two big arguments. The first one, common descent. Basically, all life came from a common ancestor that one people accepted pretty quickly.
Makes sense.
But his second argument, that natural selection is the main driver of evolution, that was a harder sell.
Yeah, I can see that. The idea that new species just kind of appear gradually because of natural selection acting on little variations, that's a lot to take in.
Definitely. And a big problem for Darwin was he didn't have a good explanation for how inheritance worked. He saw that offspring looked like their parents. He called it the strong principle of inheritance. But he didn't know the biology behind it. Later, he tried to explain it with this Jimuel's theory, but that turned out to be wrong.
Ah, so there was this gap in his theory, and that made it open to some pretty tough criticisms, right?
Right. One big problem was this blending inheritance idea from Fleming Jenkin. He argued that if traits blended together in offspring, like mixing paint, any new variation would just get diluted over time.
I see.
And that dilution, he said, would stop natural selection from having enough heritable variation to work with to make those complex adaptations. Darwin couldn't really argue against this with his limited understanding of inheritance.
So the way people thought about inheritance back then actually undermined Darwin's whole theory of natural selection. That's a big problem.
It was. But the cool thing is, inheritance doesn't actually work by blending. It works by particles. Mendelian inheritance, that's what we call it now, says that offspring inherit these separate units, what we now call genes, from their parents.
Okay.
And these units stay separate, they don't blend together. That solves Jenkin's problem, because it shows that heritable variation isn't lost during sexual reproduction. So this philosophical problem with Darwin's theory wasn't solved by new biology in Darwin's time. It was solved later by a mathematical understanding of how inheritance works.
So the variation sticks around and can be passed down over generations. But Mendel's work, his groundbreaking stuff, people didn't really get it at first, did they?
No, they didn't. His experiments with pea plants back in the mid-1800s, super detailed work documenting how traits are passed down. The scientific community just ignored it for like 40 years.
40 years. That's like a huge delay in scientific progress. What were the key things he found that turned out to be so important?
Well, with his pea plants, he figured out some basic principles. He showed that traits often come in dominant and recessive forms, like round seeds or dominant over wrinkled seeds. He also came up with the idea of segregation, which means that the hereditary factors, the alleles, for each trait separate when gametes are formed. So each parent only gives one allele to their offspring.
I see.
And when he looked at the first generation of offspring, the F1, and then the second generation, the F2, he found these predictable ratios of traits. like the famous 3.1 ratio for a single trait, which showed how inheritance works.
Yeah, the 3.1 ratio in the F2 generation, that really shows those discrete units of inheritance at work. So why was his work ignored for so long?
Well, a few reasons. The scientists back then weren't really looking for a mathematical approach to heredity. And Mendel was working on his own, kind of isolated.
Hmm.
Even after his work was rediscovered around 1900, people thought Darwin's idea of gradual evolution and Mendel's idea of these separate units of inheritance, they didn't fit together.
They didn't fit together, but it seems like they should,
Yeah,
right?
you'd think so. See, the early Mendelians, they were all about discontinuous variation, like those clear-cut traits, either or, like the seed shape.
Mm.
Some thought that big evolutionary changes happened because of big mutations in these units.
Oh, okay.
And that clashed with Darwin's idea of gradual evolution from small, continuous variations. The early Darwinians, they were used to thinking about continuous traits, like height or beak size. They couldn't see how these could be explained by Mendel's factors. So there was this tension between the suddenness of Mendelian inheritance and the gradualness of Darwinian evolution.
Two groups, each with a piece of the puzzle, but they couldn't see how to put them together. Sounds like a classic story in science.
It is. And population genetics came about from scientists trying to bridge that gap. R.A. Fisher, in 1918, wrote this super important paper. He showed mathematically how continuous traits, the ones Darwin was into, could be explained by lots of Mendelian factors working together, each one having a small effect. He showed that with enough of these factors, you'd get that normal, continuous distribution of a trait in the population.
That was the missing piece, showing that Mendelian inheritance could explain both discrete and continuous variation.
Exactly. But the real breakthrough, that came in the 1920s and 30s with Fisher, J.B.S. Haldane, and Sewall Wright. They created these mathematical models to study how natural selection and other things like mutation and genetic drift would affect the frequencies of Mendelian alleles in populations over time. This meant they could actually explore evolutionary hypotheses quantitatively.
So they took Darwin's qualitative ideas about selection and combined them with Mendel's laws of inheritance, and they turned it all into math, making it possible to test predictions about evolution at the gene level. That's amazing.
Totally revolutionized the field, moved evolutionary biology from just describing things to actually making and testing predictions with numbers. And this whole strategy, using mathematical models, it's still super important in population genetics today. Plus, as our source says, with molecular biology and gene sequencing, the field has exploded with data, tons of empirical data to test those theoretical models, which is something the pioneers could only dream of.
So it's gone from a theoretical thing to a field deeply rooted in data, and it's still refining our understanding of evolution at the gene level.
Exactly. And even in those early days, there are big debates. Fisher and Haldane, they focused on natural selection as the main driver of adaptation, right? He agreed selection was important, but he also emphasized the role of chance, specifically random genetic drift, especially in small populations. He also looked at gene flow between different populations. And this debate about how important selection is versus chance, it became a huge theme in evolutionary biology, leading to the neutralist versus selectionist controversy in the 60s and 70s. Motu Kimura argued that a lot of the genetic variation we see at the molecular level is actually neutral, meaning it doesn't affect survival or reproduction, so random drift would be the main thing shaping that variation.
And that debate, it's still going on today in various forms, right? Biologists are trying to figure out the balance between deterministic forces like selection and random forces like drift in shaping genetic diversity. Even with all the power of modern genomics, those basic questions about what drives evolution are still front and center.
Absolutely. Those theoretical foundations from the early 20th century are still incredibly relevant for understanding all the genomic data we have now. But okay, that's the historical background. Let's dive into one of the most important concepts in population genetics, the Hardy-Weinberg principle. What is it, and why is it so important?
Right, the Hardy-Weinberg principle. G.H. Hardy and W. Weinberg figured it out independently back in 1908. It's often called the null hypothesis of population genetics. It's what you'd expect to see if nothing was pushing evolution in any direction.
Exactly. It describes what would happen in a large population that mates randomly. If there's no selection, no mutations, no individuals coming in or leaving, and no random genetic drift, then the frequencies of alleles and genotypes will stay the same from one generation to the next. It's like a state of genetic equilibrium.
Okay, so to be in Hardy-Weinberg equilibrium, there are some specific conditions that need to be met. Sounds kind of idealistic, not really realistic.
You're right, the conditions are pretty strict. The population has to be massive to minimize random effects. Mating has to be totally random. And there can't be any evolutionary forces acting on the population.
Mhm.
No natural selection, no new mutations, no migration, no genetic drift. We're also usually talking about diploid organisms, the ones with two sets of chromosomes with haploid gametes,
Mhm.
sperm and egg. And we're usually looking at one locus on an autosome, that's a non-sex chromosome, just to keep things simple at first.
So yeah, super idealized. In the real world, it's pretty likely that at least one of those conditions isn't going to be true.
Definitely. But that's what makes it so useful. It's a theoretical benchmark. If we see a real population that's not in Hardy-Weinberg equilibrium, it means something's going on. Some evolutionary force is causing those allele and genotype frequencies to change.
Okay, I get it. So how do we actually express this mathematically?
All right, so imagine a gene with two alleles. Let's call them A1 and A2. The frequency of A1 is P, and the frequency of A2 is Q. Since those are the only two, plus Q equals 1, right? Now, Hardy-Weinberg says that the next generation will have these genotype frequencies. A1 will be squared, A1 A2 will be 2 times Q, and A2 will be Q squared.
So to get a homozygous genotype, two of the same allele, you just square the frequency of that allele. And to get a heterozygous genotype, one of each allele, it's twice the product of their frequencies.
Exactly. And you can expand this to more than two alleles. If you have n alleles with frequencies and so on up to PD, then the frequency of a homozygote would be pi squared. And the frequency of a heterozygote would be 2 times pi zeta.
Okay, I think I'm following. Now, you mentioned random mating. Why is that so important for Hardy-Weinberg?
Because random mating basically shuffles all the alleles in the population like a deck of cards. If mating is random, the chance of any two alleles getting together is just the product of their frequencies in the gamete pool. And if there's no selection, mutation, or migration, the allele frequencies in the gametes are the same as in the parent population. So, random pairing of gametes naturally leads to the genotype frequencies predicted by Hardy-Weinberg. Interestingly, you usually reach this equilibrium in just one generation if there's no overlap between generations, or it might take a bit longer if there is overlap. And once you hit those Hardy-Weinberg proportions, they stay stable as long as all the other conditions are met.
So it's a stable genetic situation as long as there are no evolutionary pressures. What's the big philosophical takeaway from Hardy-Weinberg?
Well, like we said before, it directly solves the blending inheritance problem that Darwin struggled with. Remember, Jenkin thought that sexual reproduction would just blend everything together and you'd lose variation. But Hardy-Weinberg shows that's not true. Sexual reproduction itself doesn't decrease genetic variation at the allele level. It just shuffles existing alleles into new genotype combinations, and it does it in a predictable way. Now, natural selection or other forces might act later to reduce variation, but sexual reproduction itself isn't the problem. So, a mathematical concept revealed a flaw in a biological argument, a classic example of how theoretical models can drive progress in science and also, in philosophy.
That's pretty cool. A theoretical framework can solve a big conceptual problem in understanding the natural world. But besides that, how is the Hardy-Weinberg principle used in practice by population geneticists?
It gives us a simplified way to model evolutionary change. If we know a population is in Hardy-Weinberg equilibrium for a specific gene, we can track its genetics just by looking at allele frequencies. That's much easier than keeping track of all the different genotype frequencies, especially with multiple alleles or genes.
You're right, it's so much easier.
It lets us focus on how those evolutionary forces cause deviations from this equilibrium, which shows us the impact of those forces.
So it's like a starting point, a model to compare real populations against so we can see if evolution is happening.
Exactly. And while you'll rarely find a perfect Hardy-Weinberg situation in nature, it's a decent approximation for some genes in some populations. Our source mentions the human ABO blood groups. In many populations, the genotype frequencies are close to what Hardy-Weinberg predicts because people don't usually choose their partners based on blood type, and the other evolutionary forces acting on these genes are pretty weak. But for other traits like height in humans, where people do tend to choose partners of similar height and lots of genes are involved, you wouldn't expect to see Hardy-Weinberg equilibrium.
So it's a useful theoretical tool, but it also reminds us that real populations are complex, with lots of things influencing them.
Exactly. The famous population geneticist W.J. Ewens, he said that Hardy-Weinberg is both one of the easiest theorems to prove and one of the most important. It's important because it shows us how genetic variation is preserved in populations that reproduce sexually. Okay, now that we understand this genetic equilibrium thing, let's look at the forces that actually drive evolution by disrupting it.
Yeah, let's get into that. These are the factors that change allele and genotype frequencies over time, causing evolution. Our source lists four main forces: natural selection, mutation, gene flow or migration, and random genetic drift. Let's start with natural selection, which is often seen as the most important one in driving adaptation. How do we model that in population genetics?
So in population genetics, natural selection means that individuals with different heritable traits, specifically different genotypes, have different chances of surviving and reproducing. The simplest model looks at a single gene with two alleles in a big population that mates randomly, just like our Hardy-Weinberg setup. But now we say that the three possible genotypes, A1, A1, A2, and A2, they have different fitnesses. Fitness, we usually call it, with subscripts for the genotype. It tells you the average number of offspring that individuals with that genotype produce compared to the population average.
So, a genotype with higher fitness is more likely to pass its alleles down to the next generation, changing the allele frequencies over time. That's like Darwin's survival of the fittest, right? How do we translate that into math in population genetics?
Well, if the fitnesses of the genotypes aren't equal, the allele frequencies will change over time in a predictable way. We use recurrence equations to describe how the frequency of an allele, let's say A1, with frequency in the current generation, is related to its frequency, which we call in the next generation.
Okay.
These equations take into account the fitnesses of the genotypes and their frequencies, which we usually assume are in Hardy-Weinberg proportions before selection happens. They basically calculate the weighted average contribution of each genotype to the next generation's gene pool based on its fitness.
So, from those equations, we can figure out the change in allele frequency from one generation to the next. What do these models tell us about how selection works under different fitness scenarios?
They give us some really interesting insights. For example, if the A1 genotype has the highest fitness, meaning W is greater than W and W2, then the model predicts that the A1 allele will become more common over time, eventually becoming the only allele at that locus, which we call fixation.
What wins out?
Exactly. And if A2 has the highest fitness, then A2 will go to fixation. This is called directional selection. It's pushing the population towards a specific genetic makeup.
So, selection can get rid of less beneficial alleles and establish the most beneficial one. But what if the heterozygote A1A2 has the highest fitness better than both homozygotes, so is greater than both W and W? What happens then?
That's a cool case. We call it heterozygote advantage or overdominance. In this situation, neither allele will necessarily be completely eliminated. Instead, the population will often reach a stable point where both A1 and A2 are maintained at specific frequencies. The math shows that there's a point where the increase in one allele's frequency due to the heterozygote advantage is balanced by a decrease when it shows up in the less fit homozygous state. This leads to a polymorphic equilibrium, where selection actually maintains genetic diversity at that locus. It shows that natural selection isn't always about making everything the same. It can also actively maintain genetic variation in certain situations.
That's a really important point for thinking about diversity and stability in biological systems. And population genetic models let us quantify the speed of these evolutionary changes too, right?
Absolutely. We can use the selection coefficients, which measure the fitness differences between genotypes, to model how fast allele frequencies change under different selection pressures. We can ask questions like, how long does it take for a beneficial mutation to spread through a population? Or, how much slower is a recessive harmful allele eliminated compared to a dominant one? This quantitative approach is a key part of population genetics, allowing us to make precise predictions about the pace and way evolution happens.
That's amazing. Okay, so we've covered selection. Now, what about mutation? It's often called the ultimate source of new genetic variation, right? How does population genetics handle mutation?
Mutation is the foundation of all heritable variation. Natural selection works with the variation that's already there, but mutation brings in totally new alleles. That's what keeps the population from becoming genetically uniform. When we combine mutation with selection, it can change our predictions about evolution. Even if one allele is favored by selection, mutations can keep introducing other alleles at low frequencies, keeping a certain level of genetic polymorphism in the population.
So there's this constant influx of new genetic possibilities, even though most of these mutations aren't beneficial. How do the models account for the interplay between mutation and selection?
We can tweak those recurrence equations for allele frequency to include mutation rate, which is usually represented by U. That's for the mutation rate from one allele to another, like A1 to A2. These rates tell you the chance that an allele will mutate to another form in a single generation.
So the change in allele frequency will be affected by both selection and the rate of mutation.
Precisely.
Interesting.
And this balance between new alleles coming in from mutation and them being selected for or against can create interesting equilibrium states. One important concept is selection-mutation balance. Imagine a harmful allele that's selected against but keeps popping up because of mutation. The frequency of that allele will be determined by how often it appears through mutation and how strongly selection is working against it.
I see.
For example, let's say you have a recessive deleterious allele with a mutation rate of U and a selection coefficient of against the homozygous recessive genotype. The equilibrium frequency, we call it Q, is about the square root of U divided by S. This means that if the mutation rate is high, or the selection against the allele is weak, you'll see a higher frequency of the deleterious allele. Our source mainly talks about harmful mutations here because beneficial mutations usually become more common pretty quickly due to positive selection. So adding another copy of an already super beneficial allele doesn't really change much in terms of the overall allele frequency.
Okay, that makes sense. Selection really drives the fate of beneficial mutations, while for the harmful ones, it's all about balancing with the mutation rate to see how common they are in the population. All right, what about the third force, migration or gene flow between populations? How do we model that?
Migration is when individuals move from one population to another, right? And if those migrants have different allele frequencies than the residents, the genetic makeup of the population they join can change. We model this using a migration rate, usually called M, which is the proportion of migrants in a generation. And we also use the allele frequency of the migrant population. The change in allele frequency in the receiving population is proportional to the difference between the resident allele frequency and the migrant allele frequency, and it's also proportional to the migration rate.
Okay, so if the migrants have more of a certain allele, it'll become more common in the population they join, and vice versa. Sounds like it would make allele frequencies more similar between connected populations.
Exactly. Gene flow is like mixing the genetics of different populations. It reduces the genetic differences that might happen because of things like selection acting differently in different environments, or genetic drift in isolated populations. Even a little bit of migration can counteract those diversifying forces and keep populations genetically similar. This homogenizing effect of migration is important for things like speciation and how species adapt to their local environments. It also plays a role in the debate about group selection, because for selection to work at the group level, you usually need significant genetic differences between groups.
So, migration can both bring in new genetic variation and stop populations from becoming too different. Right, the fourth force, random genetic drift. This one seems different, more about the role of chance in evolution. How do we approach this in population genetics?
Random genetic drift is basically random fluctuations in allele frequencies from one generation to the next, and it happens because populations are finite, meaning they have a limited number of individuals. Remember how in our models for selection and mutation, we assumed an infinitely large population?
Right.
Well, in real life, all populations are finite, and especially in small populations, these random sampling effects can really drive evolution.
Interesting.
An allele's frequency can go up or down just by chance. Like, if individuals carrying that allele happen to have a few more offspring in one generation, it's not because the allele makes them fitter, it's just random. This is where chance really comes into play in evolution, making things a lot less predictable.
So it's like picking a small sample from a big pool. The proportions in your sample might not perfectly match the original pool just because of random chance. And over many generations, these small random changes in allele frequencies can build up and cause big evolutionary changes, especially in small populations.
Exactly. There's the narrow definition of drift, which is specifically about this random sampling of gametes during reproduction. And then there's the broader definition, which includes all the random factors that can affect allele frequencies. We're focusing on the narrow definition here. With drift, it's harder to predict exactly what the genetic makeup of future generations will be. Instead of definite outcomes, we talk about probabilities, like the probability distribution of possible allele frequencies.
So instead of predicting one outcome, we're predicting a range of possible outcomes and how likely they are. What's a good model for studying random drift?
The Wright-Fisher model is a classic one. It imagines an idealized population of N diploid individuals, meaning they have two sets of chromosomes with non-overlapping generations. They mate randomly, and there's no selection, mutation, or migration. In this model, the next generation is created by randomly choosing two N gametes from the current generation's gene pool. The number of copies of an allele in the next generation follows a binomial probability distribution based on its frequency in the current generation.
So what happens in the long term because of this random drift?
One major outcome is that one allele at a locus can become fixed, meaning it reaches 100% frequency, while the other alleles at that locus are lost, their frequency drops to zero. These states of fixation and extinction are like endpoints in the model. Once an allele is fixed or lost, it'll stay that way unless a new mutation comes in or migration brings back a different allele. Because of this tendency toward fixation and loss, random drift actually makes a population genetically more uniform over time. It reduces variation.
So mutation adds diversity and drift takes it away, especially in small populations. What determines whether an allele will become fixed or lost due to drift?
If an allele is neutral, meaning it doesn't affect fitness, the chance of it becoming fixed is simply equal to its current frequency in the population. So a rare allele has a small chance of becoming fixed and a big chance of being lost. Imagine a new neutral mutation that appears just once in a diploid population of size n. Its initial frequency is 1 divided by 2n. That means its chance of becoming fixed due to drift is also 1 divided by 2n, which is a tiny probability in any decent-sized population. This shows us that most neutral mutations are lost by chance.
That makes sense. Now, you said that's for neutral alleles. What happens when you have natural selection and random drift working together in finite populations?
That's when things get interesting. The fate of an allele depends on the relative strengths of selection and drift. There's a key parameter, 4 times need times. Need is the effective population size, which might be different from the actual number of individuals because of various factors, and is the selection coefficient, which tells you how much of an advantage or disadvantage the allele gives. If 4 times need times is a lot bigger than 1, selection is in control, and a beneficial allele will probably become fixed, while a harmful one will probably be lost.
Okay.
But if 4 times need times is a lot smaller than 1, then random drift is the boss. And even a slightly beneficial allele might be lost by chance, or a slightly harmful one might become fixed. This whole interplay between selection and drift was a big part of that neutralist-selectionist debate we talked about earlier. Neutralists argued that for a lot of genetic variations, the selection coefficients are so small that drift is the main force behind their evolution.
So population size is key for figuring out if selection or drift is more important. In small populations, chance can even override weak selection. Our source also talks about hitchhiking, or genetic draft, in the context of drift. What's that all about?
Hitchhiking or genetic draft happens when a neutral or even slightly harmful allele becomes more common, just because it's linked on the same chromosome to another allele that's being strongly favored by selection. So this neutral allele gets carried along by the selected allele, even though it doesn't actually help the organism survive or reproduce. It's another way allele frequencies can change randomly, not because of selection on that specific allele, but because of its association with a selected locus.
Like riding someone else's coattails. Interesting. Our source also points out that there's some debate about the whole concept of random drift, philosophically speaking. What are the main points of disagreement?
Well, some philosophers of science, like Roberta Milstein, have said that the term random drift is used a bit loosely. Sometimes it refers to the random sampling of gametes, and sometimes it refers to the result, the change in allele frequencies. This has led to discussions about how to define drift properly and how it's different from natural selection as a cause of evolution. It raises some fundamental questions about what causes evolution and the role of chance in shaping the history of life.
So even a seemingly simple concept like random drift gets pretty complicated when philosophers start poking at it. Okay, so we've been talking about evolution looking forward from one generation to the next, but our source then brings up coalescence, which is a way of looking backward in time. What's that all about?
Coalescent theory gives us a different perspective. It starts with a sample of genes from a current population and traces their ancestry back in time. The basic idea is that if you trace the lineage of any 2 copies of a gene back through time, they will eventually meet at a single common ancestor, called their most recent common ancestor, or MRCA. Coalescence is the event where those gene lineages merge as we go further back in time.
Like tracing family trees, but for genes. Instead of looking at your descendants, you're looking at the ancestors of genes, going back to their common origin.
Exactly. Coalescent theory uses probability models, often based on that Wright-Fisher model we talked about, to estimate the probability that 2 randomly chosen gene copies from the current population shared a common ancestor at some point in the past. The rate of coalescence mainly depends on the effective population size. In smaller populations, genes coalesce faster because there are fewer possible ancestors.
Makes sense. So, what can we learn about a population's history by using this backward-looking approach?
Coalescent theory is really powerful for figuring out things about a population's past. Things like how big the population was historically, how individuals moved between populations, when populations split, and even the effects of past selection events.
Wow, so much information.
Yeah, it's amazing. By analyzing the patterns of genetic variation in a sample of DNA and comparing them to the predictions of coalescent models under different scenarios, we can understand the population's evolutionary history in a way that's often not possible just by looking forward in time. It also helps us understand patterns of DNA variation within and between species.
So, looking at genetic relationships in the present can help us reconstruct the evolutionary past. Our source mentions that the average time it takes for two genes to coalesce is about 2n, where n is the effective population size. That seems like a long time for large populations.
It can be. It means that gene lineages can stick around for a long time before they happen to coalesce. But remember, that's just an average. There's going to be a lot of variation. Some gene pairs will coalesce much more recently, and some will have a common ancestor much further back. And the cool thing is that coalescent theory can be extended to deal with more complicated situations, like multiple alleles, recombination, population subdivision, and changes in population size over time. It's a super versatile and widely used tool in population genetics and phylogenetics today.
It's an incredible tool for understanding the evolutionary history that's written in the genomes of living things. Okay, our source then switches gears and talks about some criticisms of population genetics, questioning its place within biology as a whole. What are the main concerns people have raised?
Well, over the years, there have been a few main criticisms. One is that population genetics has been too focused on developing abstract mathematical models, sometimes without enough focus on testing those models with real data and connecting them to real biological systems. Critics have said that this theoretical focus has sometimes led to a gap between the elegant math of population genetics and the messy reality of how traits evolve and how organisms interact with their environments. However, with all the molecular data we have now, this criticism is less common because molecular population genetics is now very much involved in analyzing huge genomic data sets.
So the field has become more data-driven, which addresses that concern about not being grounded in reality. What are some of the other criticisms?
Another common one is that the models in population genetics are too simplified. They often make assumptions that aren't true in real populations, like assuming large, randomly mating populations with no environmental variation. Critics argue that this makes it hard for those models to accurately reflect the complexity of real populations and evolutionary processes, especially when we're trying to understand how complex traits evolve. They say that focusing on changes in single gene frequencies in these idealized scenarios doesn't capture the dynamics of traits that are influenced by many genes and the complicated relationship between genotype and phenotype. There's also this historical argument that even though population genetics is theoretically important, it hasn't.